Greetings to everyone. Welcome back once again to my educational channel on biology. Teacher Janet here. So in this video, I'm going to discuss activity 3.1, which is related to diffusion across the Visking tubing, a very hot topic, right? So in this video, it's going to be a bit different because I'm going to give you some soalan, ramalan, or forecast questions, okay, that are usually found in exam papers, like what is the observation, the inference, hypothesis. We'll continue to discuss operational definition with new questions and then methods to handle the variables, prediction questions, categorization or classification questions. All right. So all this will be discussed here. We'll first discuss the experiment, the procedure, and also the background knowledge related to this activity. All right. So this will help to prepare you for the exam. So let's get started. Now, our aim is to study the movement of substances across a selectively permeable membrane, and the two substances involved are glucose and starch. Now, in the previous videos on the trial papers, I have not discussed this question before, or this experiment before, where glucose is involved, okay? Not iodine solution in the distilled water, okay? But glucose in the Visking tubing, okay? So this is a different question. Now, so we are going to study the movement of substances such as glucose and starch, whether they can move across this Visking tubing, which is a selectively permeable membrane. All right. So in this picture, you see a representation of what's happening in the Visking tubing. Starch molecules that are bigger, right, cannot go through or pass through the pores in the Visking tubing, which are smaller than the starch molecules. Whereas glucose molecules that are smaller than the pores of the Visking tubing can pass through and diffuse out of the Visking tubing. Okay, so let's go on further. So here's some background knowledge about these diffusion experiments. So firstly, the iodine test for starch. Procedure. Put about 2 ml of the sample solution in a test tube, then add a few drops of iodine solution, and then mix and observe the color change, right? So when the iodine and the sample solution is mixed together, if after you mix it, you get a yellowish brown color, this is the color of the iodine solution. So there's actually no change in color. It means that no starch is present, right? But if after you have mixed it, from yellowish brown, it turns to blue-black color, that means starch is present, okay? So in the results or the observation, always write down the color, and it's quite standard. For the iodine solution itself is yellowish brown in color, not brown, not yellow, yellowish brown, remember that, okay? And that's the negative result for starch. That means no starch is present. But if starch is present, you write it as blue-black color. That's the final color. Don't write blue, don't write black, write blue-black color, okay? Now next, the Benedict's test. So Benedict's test for reducing sugar. Add 1 ml of Benedict solution to 2 ml of the sample solution in a test tube. This is following the textbook. Now I find that sometimes we have from experience. Sometimes if you just add 1 ml Benedict solution to 2 ml of the sample solution, 1:2, sometimes you may not get a very obvious result. So if you are expecting the glucose to be, the reducing sugar to be outside the Visking tubing and you cannot get a positive result, try adding more Benedict solution. All right, if you cannot get it and you expect a positive result for reducing sugar. Okay, now so you may have to increase the amount of Benedict solution used. Now heat the test tube in a boiling water bath. After you have mixed the two solutions together, Benedict solution and sample solution, you must heat it in the water bath like this for a few minutes. Okay, then you see the gradual change. If reducing sugar is present, the solution changes color from blue to green to yellow, orange, and finally a brick red precipitate is formed. Now what if the solution is green or yellow or orange and it stops there? It didn't become a brick red precipitate. Is reducing sugar present? Yes, it's still present, but in smaller amounts and smaller concentrations. Okay, it's still a positive result if you get a green color or yellow or orange color, even if there's a slight change in the color of the mixture. Okay, but if reducing sugar is absent, the solution does not change color at all, it will remain blue. Don't write does not change color, just write blue. Okay, if it's a negative result for reducing sugar, just write down that it's a blue color. Okay, that's the color of the Benedict's solution. Now, continuing the Benedict's test for reducing sugar, the procedure based on textbook, as we have mentioned just now, okay, is a repeat here. Place 2 ml of sample solution in the test tube and 1 ml of Benedict's solution. You may need to add more for better results, but in the exam, you are asked to explain the procedure, then you follow the textbook. Okay, 2:1, 2 ml of sample solution to 1 ml of Benedict's solution. Heat the mixture in the boiling water bath for 2 to 5 minutes and observe the color change. Now, results. If blue, that means no color change, no reducing sugar present, like this one here, right? Now, if it's green color or orange, yellow or orange, there are some amounts of reducing sugar. And of course, if it's brick red precipitate, it means there's a higher concentration of reducing sugar. All right? So even a green color or yellow or orange color is a positive result for presence of reducing sugar. So what are the examples of reducing sugars? Glucose, fructose, galactose, lactose, and maltose. They reduce copper 2 ions to copper 1 ions. Okay, copper 2 ions in the Benedict's solution to copper 1 ions, and that's how the brick red precipitate of copper 1 oxide is formed. Okay, now examples of non-reducing sugar, there's only one example here, is sucrose. It does not reduce copper 2 ions in Benedict's solution, so the Benedict's test will be negative if you mix Benedict's solution with sucrose and you heat it up. The solution will remain blue. Don't say no color change. I'll just say it remains blue or blue color. Now, see, for classification questions in the exam, you can be asked to classify the molecules according to whether they can diffuse through the Visking tubing or whether they cannot. Okay, so molecules can diffuse through the Visking tubing or that are small enough to pass through the pores of the Visking tubing include water, glucose and fructose, which are monosaccharides. They are small molecules. Iodine molecules, I2. Amino acids, which are small building blocks of proteins. They can pass through the Visking tubing. And charged ions like sodium ions, chloride ions, salt, that means salt, and potassium ions, okay, small charged particles. How about molecules that cannot diffuse through the Visking tubing because they are too big, too large, or complex to pass through the pores? These include starch, which is a large polysaccharide, so it's blocked from diffusing through the Visking tubing pores. Sucrose, which is a disaccharide, so it's big compared to the glucose molecule. Proteins, which are macromolecules, and DNA, which is a large nucleic acid polymer. Now, for classification questions also, you can be asked to classify, okay, membranes into selectively permeable membranes or non-permeable membranes. Or they can ask you to give examples other than Visking tubing. What are other examples of selectively permeable membranes? Okay, so these are all the selectively permeable membranes examples. Visking tubing, plasma membrane, egg membrane, which is a white membrane beneath the shell, okay, behind the shell. And then the raw potato plasma membrane. So it's still plasma membrane. That means you have a raw potato. The cells contain plasma membrane, and those are all selectively permeable membranes. Okay. Nuclear membrane in the cell allows RNA to move in and out, move out of the nuclear membrane. There's pores, so it is a selectively permeable membrane. Non-permeable membranes include the eggshell itself, or the best example is a rubber sheet, okay, or plastic wrap. And also boiled potato plasma membrane. The potato is boiled. The plasma membrane structure has been broken down, and the heat has denatured the proteins in the plasma membrane. So the membrane will lose its function, and there's no osmosis, and it cannot act as a selectively permeable membrane. So these are the two different experiments that we have discussed so far. The SPP paper 3, where we have we put iodine solution in the distilled water outside the Visking tubing and starch inside the Visking tubing. Okay, so at the end of the experiment, we'll find that the iodine has moved into the Visking tubing and caused the mixture, the starch, to turn to blue-black color, right? Okay, so that is the one that we discussed, experiment we discussed earlier. So in this experiment, it's different. So we're going to talk about the content inside the Visking tubing, which is different. It contains glucose and starch, and outside there's no iodine in the distilled water. Okay, so this is activity 3.1. So now let's discuss activity 3.1. To study the movement of substances across a selectively permeable membrane. So our substances here are glucose and starch, and the selectively permeable membrane is the Visking tubing. Now let's look at the the various aspects. Studying the movement of substances across a selectively permeable membrane, such as the Visking tubing. Problem statement is, now problem statement must have your MV and RV. So straight away from your activity 3.1 or from the question in the SPM paper, you have to select your MV and RV in the aim. Okay, so movement of substances is your MV. The MV is the substance. Okay, the substance. And the movement of substances, whether they can move through, is the RV. Okay, substances is the MV, the types of substances with different sizes of molecules, and your movement, whether they can move through the selectively permeable membrane, is your RV, your main RV. Okay, so problem statement: does the size of the dissolved particles or molecules affect the movement of substances across a selectively permeable membrane? Problem statement is always a question. Okay, so here you still have your MV and RV. So the movement of the substances, that is the RV, and the size of the dissolved particles or size of molecules is your MV. Now let's go on to hypothesis, which still has the MV and RV, the main MV and the main RV. Only small molecules are able to diffuse through a selectively permeable membrane, while large molecules cannot diffuse through a selectively permeable membrane. Okay, so small molecules and large molecules, this is your MV. All right. Now, able to diffuse and cannot diffuse, this is the RV. That's all. And the relationship is that small molecules can diffuse through the selectively permeable membrane, like Visking tubing, whereas larger molecules cannot diffuse through. Okay? So MV, RV is all here in the aim, problem statement, and hypothesis. Now let's go on to variables. Your manipulated variable is only one. It's the size of the molecule, or molecule size. Okay, such as the size of the glucose molecules and the size of the starch molecules. So very simple. There's no sub MV at all. Okay, it's just one MV. Now, responding variable, there are few responding variables. Some are sub. Now, the main one is the ability of the molecule to diffuse through the selectively permeable membrane. The ability. But in your experiment that you're doing, you want to find out whether the molecules can move through the Visking tubing into the beaker. Then you have to test for the presence of these molecules in the Visking tubing, and especially in the beaker, after 30 minutes or 15 minutes. So that becomes your sub-RV. And to test that the presence of molecules, you have to use the iodine test and the Benedict's test. So that the color change in the iodine test and the Benedict's, the iodine test and the Benedict's test is also a sub-RV that you observe in the experiment. Okay, but here they just generally put as presence of molecules. Okay, but to test for the presence of molecules, you have to look at the color changes in the iodine test and in the Benedict's test. Now, fixed variable is the surrounding temperature and soaking time. So we are going to use this as a fixed variable and not suggest other examples which are actually not suitable as fixed variables. All right. Soaking time means soaking time for the Visking tubing, which is 30 minutes in this experiment, but in the, if you are doing for an exam, practical exam, it should be 15 minutes, about 15 minutes. Cannot be 30 minutes because it's too long. Okay, take note now. For exam, it's 15 minutes. Now, materials. Benedict solution, 1% starch, suspension, iodine solution, 30% glucose solution, distilled water, Visking tubing, 12 cm in length and thread, and you have a beaker, test tube, Bunsen burner, dropper, and measuring cylinder. Now let's look at our procedure. Now procedure should contain method to handle the MV, method to handle the RV, and then the FV plus the precautions. All right, even when you are writing the procedure. Now, so let's say, in this case, maybe the procedure is given. Depends on whatever it is, we still need to know the procedure well. Okay, so firstly, soak the Visking tubing in water for 5 minutes to soften it. Okay, so once you're ready to start the experiment and given the time to start the experiment, let's say after reading and checking your apparatus, then they say you can start. You better soak your Visking tubing first of all, okay, for a few minutes. Then make a knot and tie one end of the Visking tubing tightly with a thread to prevent leaking. Okay, the key word here is tie it tightly. Okay, tie one end tightly with a knot, a tight knot. Now, you can see this experiment being done in one video, all right, which I put the link below. Okay, so see the link below to see how they do the experiment. All right, how they carry out this experiment. It's good for you to look at that video to revise, okay, the practical aspects of how to carry out the experiment. Okay, so if this is written down, procedure is for you to write down, and you must write down, tie one end of the whisking tubing tightly with a thread to prevent leaking. Next, fill the whisking tubing with 10 ml glucose solution and 10 ml starch suspension by using a syringe. So this is your MV. You use different solutions or substances with different size of molecules, okay, such as glucose and starch. This is the way to handle the MV. Use different solutions or different substances with different size of molecules, such as glucose and starch molecules. Tie the other end of the whisking tubing tightly with thread. So you have poured in your two solutions, that is glucose and starch suspension, and then glucose solution and starch suspension, and then you tie this end, the top end, tightly with a thread. Okay, all right. Now the color of the solution is recorded. So basically the color of this solution, because of the starch suspension, it will be cloudy a bit, white and cloudy, white cloudy solution. Okay, tie the other, so the color of the solution is recorded, and then next, rinse the outside of the whisking tubing with distilled water. All right, so this is a precaution. That means before you soak the whisking tubing or immerse it in the distilled water, rinse the outside with distilled water at the sink. Rinse it to get rid of traces of starch or glucose. Then add 400 ml of distilled water in the beaker, right? Then place the whisking tubing in the beaker and leave for 30 minutes. Okay, now in your exam, I don't think you can leave it for 30 minutes, maybe 15 minutes or so. Now after 30 minutes, transfer the whisking tubing into a dry beaker, all right, and then you conduct the two tests, the iodine test and the Benedict's test. Now, so this precaution, the one where you rinse the outside of the whisking tubing with distilled water, is quite important. In case some of the starch from your syringe has spilled onto the outer layer of the whisking tubing, outer part. Okay, so make sure you rinse the outside of the whisking tubing carefully with distilled water before you put it into the distilled water and leave there for, say, 15 minutes, or in this case 30 minutes. Okay, now next, take out the whisking tubing after 30 minutes or 15 minutes, then you transfer it to a dry beaker, and you're going to open up the whisking tubing, all right, and conduct the iodine test for solutions in the whisking tubing. And also, whatever is left in the beaker, you have to also test. This is also important. Don't simply spill all the whisking tubing contents out into the distilled water at the end of the experiment. Take out the whisking tubing very carefully, and this distilled water here, you're going to test for the presence of starch and glucose also. Okay, and then the whisking tubing, you're also going to test for the presence of starch and glucose. All right, so conduct the iodine test for the solutions in the whisking tubing and in the beaker. Now this is the iodine test. Put 2 ml of each solution in separate test tubes and add 1 ml of iodine solution and observe the color produced. Okay, that is the RV, the color change when you carry out the iodine test. What is the color produced in the iodine test, right, or in the iodine solution after you have mixed it with a sample solution? Now next, conduct the Benedict's test on solutions in the whisking tubing and in the beaker. Again, this is the test, so you will be asked to explain the test. All right, so you're expected to know the steps. So put 2 ml of each solution into separate test tubes and add 1 ml of Benedict's solution. Okay, heat the solutions in the water bath for 5 minutes and record any changes in color. Remember, you have to heat the solution. Okay, 2 ml of the sample solution with 1 ml of Benedict's solution, then heat in the water bath for 5 minutes and record any changes. So the color change for the Benedict's solution is also one of the RV. All right. Now this table of results is from your textbook. Let's analyze it. Does it show MV and RV? It does show the RV, but not the MV. Okay, because it just shows you the location, the whisking tubing and the beaker. Okay, and what's the initial contents. Actually, the MV are the two substances, glucose solution and starch suspension. Okay, so this is not a table showing you MV and RV. If you want to write the MV, you should write the glucose solution as one of the substances here, and then starch suspension below. All right, so anyway, you can see the RV here, the sub RV. These are the sub RV, And what we are more interested in is what is in the water, in the distilled water in the beaker. What is the solution? What does the solution in the beaker now contain at the end of the experiment, after 15 or 30 minutes, right? Now we start with the whisking tubing first. Okay, so actually this is not the important part, all right? Now in the whisking tubing, after 15 to 30 minutes, when we do the iodine test, we find that when we mix the iodine, take a sample from the whisking tubing, a bit of the sample solution, and then add a few drops of iodine to it, you will see that you still get a blue-black color, meaning that the starch is still present in the whisking tubing, as expected, because it was initially in the whisking tubing. Okay, the starch was initially in the whisking tubing. And we test the whisking tubing content for the presence of glucose. Glucose will still be there. All right, not all the glucose diffuse out from the whisking tubing. So you get a brick red precipitate formed when you take a bit of the sample from the whisking tubing, the content, and then you add it to 2 ml of Benedict's solution, say, okay, 2 ml of the sample solution from the whisking tubing and 2 ml of the Benedict solution. Then you mix it up and then you heat it in the water bath. You will still get a brick red precipitate because initially there was glucose. So finally at the end, you still get the glucose. This is not the important part of the results. The important part is this one. What is found in the water in the beaker? Initially, there was just distilled water in the beaker. So after 15 to 30 minutes, did any of these substances diffuse out? Glucose molecules or starch molecules? Did they diffuse out? So we conduct the iodine test and the Benedict's test to find out. Okay. So now for the water in the beaker or the solution in the beaker, with the iodine test, a yellowish brown color is observed. That means there is no change in the color, right, of the iodine solution. But don't write no change in color. You must write the color that you see. It's yellowish brown color is observed. This shows that there is no starch present. If starch is present, it will be a blue-black color, right? Now, because you don't get any starch in the water in the beaker or in the solution in the beaker, that means the conclusion is starch molecule was too big and it has not diffused out of the whisking tubing into the water in the beaker. Okay, the big starch molecule has not diffused out through the whisking tubing into the water in the beaker. Now, the most important result is this one. The Benedict's test conducted on the water in the beaker, outside the whisking tubing, okay? Or the solution, conducted on the solution outside the whisking tubing. Test it with the Benedict's solution. Add 2 ml of the water from the beaker with 1 to 2 ml of Benedict's solution, mix it up, and then heat it in a water bath. You get a brick red precipitate form, okay, or maybe an orange or yellow color. Still shows the presence of glucose or reducing sugar. Okay, so if there's reducing sugar, it's obviously glucose, right? Because there's no other reducing sugar, okay, in this experiment. So glucose is present when you get a brick red precipitate, and this shows you that the glucose molecule has diffused out through the whisking tubing from inside the whisking tubing into the water in the beaker. All right, because it's small enough to diffuse through the pores of the whisking tubing. So these two, you can compare the two results here. Okay, the big molecule cannot diffuse out of the whisking tubing, but the small molecule like glucose can. Now discussion. What molecule is found in the whisking tubing, A in the whisking tubing and B in the beaker at the end of the experiment? Okay, now these are all textbook questions, so we have to go through them. Now, molecule found in A, whisking tubing, at the end of the experiment, it was not mentioned in the textbook, but it should be at the end of the experiment. Okay, so at the end of the experiment, which molecule will be able to diffuse out of the whisking tubing and which will be left in the whisking tubing? Now, the molecule found in the whisking tubing will be the starch molecules and the glucose molecules. Some glucose molecules may have diffused out into the distilled water in the beaker, right? But some are left inside the whisking tubing. So starch and glucose molecules are found in the whisking tubing at the end of the experiment. But outside in the solution in the beaker or the distilled water in the beaker, there are only glucose molecules. Okay, they are found in the solution in the beaker outside the Visking tubing. Second question, what are the inferences that can be made based on the size of the starch molecule and B, the size of the glucose molecule compared to the pore size in the Visking tubing? So let's understand this question. Inference made about the size of the starch molecule compared to the pore size in the Visking tubing. Is the size of the starch molecule bigger or smaller than the pore size in the Visking tubing? Yes, it is bigger. That's why it could not come out from the Visking tubing. So the size of the starch molecule is bigger than the pore size of the Visking tubing. Okay, that's why the starch molecules cannot diffuse out of the Visking tubing. All right, so this is your inference or the explanation for the observation. This is the inference or explanation for the observation, where you found that there was no starch present in the beaker, okay, in the distilled water in the beaker. Now, the size of the glucose molecule is smaller than the pore size of the Visking tubing. Thus, the glucose molecules can diffuse out of the Visking tubing into the beaker, and then when you test it with the Benedict's solution, you get a brick red precipitate to show the presence of glucose in the solution in the beaker, which was formerly the distilled water. Okay. Now, what are the similarities between a Visking tubing and a plasma membrane? Similarities. So both are selectively permeable membranes. Okay, and then both of them allow only certain molecules of small size to move freely across them, whereas molecules of bigger size cannot move across the membranes. This is the definition for selectively permeable membrane. Please take note of it. You can be asked to define it operationally also, right? So both of them allow only certain molecules of small size to move freely across them, whereas molecules of bigger size cannot. So molecules of small size that can move freely across them are the glucose molecules, and the bigger ones are the starch molecules. Now, conclusion for this report. The hypothesis is accepted. Only small molecules are able to diffuse through a selectively permeable membrane, while large molecules cannot diffuse through a selectively permeable membrane. Now let's look at some soalan ramalan or forecast questions or paper 3 questions that can be asked. Of course, it will be based on observation, hypothesis, inference, and so forth. So first, let's discuss the observation. State the observation for the Benedict's test on the solution in the beaker after 30 minutes. So read carefully for the Benedict's test. So the Benedict's test is this column on the solution in the beaker. So this one, solution in the beaker after 30 minutes, okay, or after 15 minutes. That means at the end of the experiment. All right. So just state the observation, meaning you must mention the Benedict's test, the solution in the beaker. So after 30 minutes, the solution in the beaker forms a brick red precipitate when tested with the blue Benedict solution in the Benedict's test. Okay, right, complete an observation. So your brick red precipitate is your sub RV. The solution in the beaker actually is the glucose, is the MV. All right, that's present to form cause the brick red precipitate to form. Okay. Now, second question, state an inference for your observation or explain your observation. They mean the same thing because an inference is an explanation for the observation. All right. So try and answer these questions first to test your skills, and then you look at the answer. Right. Let's look at the first answer. Okay. Inference. Now, the inference is for the first observation on the solution in the beaker, right? So it's about the glucose molecules and the brick red precipitate. So first inference is that we know that glucose molecules are found outside the Visking tubing. They have diffused out. So it's because the glucose molecules are small in size, so they are able to diffuse through the Visking tubing and enter the solution in the beaker. All right. Another point is how does it form a brick red precipitate? It's the reducing sugar that reduces the copper 2 ion in Benedict's solution to copper 1 ion, and then that forms a brick red precipitate. But the first 2 points are the more important ones because it's related to the experiment directly, P1 and P2. Okay. Now next, state the hypothesis for this experiment. Okay, what's your hypothesis? Let's look at the answer. It's the same as what we have discussed in the experiment just now. Only small molecules are able to diffuse through a selectively permeable membrane. Okay, small molecules you can put in brackets glucose, while large molecules, brackets starch, cannot diffuse through a selectively permeable membrane. You can put the glucose in small, in brackets, okay, small molecules glucose and large molecules starch. Now next are the variables. So of course they will not ask you for all the variables and all the methods to handle the 3 variables. They may select one, okay, but I give you the complete answer for 3 variables and 3 methods to handle the variables. Okay, so if you have not done this, you can try to write it out on a piece of paper and compare the answer. Okay, so that we can go faster here. So the variables, the manipulated variable is the size of the molecules, all right, such as the molecules of starch and glucose. So the method to handle the variables, remember, you use the word use different. So use different substances or solutions that have different sizes of molecules. I think it's better to say use different substances, because starch is not a solution, it's a suspension actually. So use different substances that have different sizes of molecules, such as starch and glucose molecules. Okay, next, the responding variable is the color change in the iodine test or solution and in the Benedict's test or solution. For the solution in the beaker, because we tested out the solution in the beaker, that is the important one, not the test for the content in the Visking tubing. That is not so important. It's for the solution in the beaker. That's the important one. So observe and record. So these words must be there. Now you cannot measure in this case, you just observe with your eyes. Okay, so observe and record the color changes of the iodine solution and Benedict's solution for the solution in the beaker at the end of the experiment or after 15 or 30 minutes, depending on how long the time is, okay, for the experiment. So in this case, we follow from the sub RV. The real sub RV here is the color change, okay, the one that you used or you recorded from your experiment is the color change. Okay, for example, that the solution in the beaker form a brick red precipitate with Benedict's test. That is the color change, right? So observe and record the color changes of the iodine solution and Benedict's solution for the solution in the beaker. If for the Visking tubing, it's not that important, because we want to see what are the molecules that have diffused out into the solution in the beaker, or the distilled water, into the distilled water in the beaker. Okay, right. So I feel that this is the best answer, all right, and based on experience, because I've carried out a lot and gone through a lot of practicals for many years, okay, done a lot of paper 3 questions too. Now, controlled and fixed variable. According to your textbook, is the soaking time, you can write there for the Visking tubing, or is the surrounding temperature. So let's look at how to write. Now, I feel the best one is soaking time, because there's a fixed time that is stated in the procedure, okay? So fix the soaking time, use the word fix, fix the soaking time of the Visking tubing at 30 minutes, or it could be 15 minutes if it's from, in the exam, they cannot let you soak for 30 minutes, it's too long, okay? So for 15 minutes or whatever time is stated in the procedure, all right, write that down. So you have to highlight the fixed variable in your question. Make sure you know, in case they ask you for the method to handle the fixed variable. Now, how about surrounding temperature? Now, for surrounding temperature, it's not so clear because there is no, you didn't go and measure the temperature in the lab, right? Okay, so if you were to write that, I would suggest you just write fix the surrounding temperature at room temperature, which is about 28 degrees Celsius. All right, fix the surrounding temperature of the experiment, for the experiment at room temperature, which is 28 degrees Celsius. Okay, but I would prefer you to write this one down. Okay, the soaking time for the Visking tubing in the beaker, right? Okay, so remember, for responding variable, write down the sub RV or operational RV. This is called the operational RV, the one that you measured or observed in the experiment. Okay, operational means experimental. All right, don't go and write what presence of molecules. All right, not so clear. Okay, presence of molecules, although that is a responding variable too. All right, the color change is the one we want to observe. Now next, operational definition. So here we come again to the operational definition for the term diffusion, all right? But it's not the same like for the SPP paper 3, because here we did not use iodine, we use glucose. We put the glucose in the Visking tubing, right? So it's different. So here again, for your components of operational definition, think of the word DOREM. D is for the basic definition. O is for all the organisms or solutions or materials that were used. You have to mention that because it's in the experiment. Okay, operational definition means experimental definition based on your experiment. And thirdly, you must write it is shown by or determined by the sub-RV in the experiment. Okay, and the RV is affected by MV. Okay, or the process of diffusion is affected by MV, which is the size of the glucose and starch molecules. Okay, so let's piece it all together. Now, the first component is the definition, but you must add in all the organisms, solutions, or materials you use. So diffusion is now movement, right? Diffusion is the movement of molecules. So this is taken from your basic definition. And then you mention the molecules used. So diffusion is the movement of molecules such as glucose, right? But not starch, because starch did not diffuse out of the Visking tubing. So I just add it in because you have to add it in. So diffusion is the movement of molecules such as glucose, but not starch. Okay, add this in. Across a Visking tubing, down the concentration gradient. So in the standard definition for diffusion in the textbook, they did mention down the concentration gradient. Okay, to make this operational definition more valid. Okay, now it is shown or determined. You can write and it is shown. Okay, and shown or determined by the final color or the color change of the solution in the beaker during the iodine test and Benedict's test, or in the iodine test and Benedict's test, after 15 minutes or 30 minutes, after the experiment, okay, or at the end of the experiment, right? So we want to look at the color, whether you know that the solution turned blue-black for iodine test, or whether the solution formed a brick red precipitate when heated with Benedict's solution. So that is your RV, your sub-RV, okay? And it is affected by the size of the, now mention the type of molecules, it's a glucose and starch molecules. If you want to be general, it's just say it's affected by the size of molecules, okay? But if you're not sure, just write down, okay? Like in the previous SPP paper, I just put it's affected by the size of molecules. You can put it's affected by the size of molecules such as P and Q molecules. Okay, write that down. Specific a bit, all right? But because the term is diffusion, it's just a general term, so we just write it like that, okay? Right, I believe this is the most complete definition for diffusion. It's 2 marks, so they can pick any two of the sentences. Now, let's go on to the prediction question. A similar experiment is carried out, but this time glucose is placed with a solution of honey in a Visking tubing. Predict the outcome at the end of the experiment when the solution in the beaker is tested with Benedict's solution. So the glucose in the Visking tubing is replaced with a solution of honey. What happens? Can you try to predict the outcome? Okay, take note. Honey in your textbook is mentioned, honey or madhu, in textbook chapter 4, page 79 in the digital version. It is said that fructose is the sugar found in honey and some sweet fruits. Okay, for grape juice, it contains a lot of glucose. Okay, but some other sweet fruits and also honey, it has fructose in it. Okay, or maybe some other sugars, but it has fructose. So we can mention about fructose. So the prediction is that because honey contains fructose, the fructose is a monosaccharide like glucose, so it can diffuse out of the Visking tubing, all right, through the pores of the Visking tubing. The size of the molecule is small. So it will form a brick red precipitate when heated with Benedict's solution, because it is a reducing sugar also, okay? So the prediction is that if it's tested with Benedict's solution, a brick red precipitate is formed when the solution is heated with Benedict's solution. Explanation. Honey contains fructose molecules. They are small in size. And the fructose molecules are able to diffuse through the Visking tubing into the beaker. So remember, we always talk about the small size and that is able to diffuse through the Visking tubing. Okay, same for glucose molecules. So the third point is related to the test. fructose is a reducing sugar that reduces blue copper 2 sulfate in Benedict's solution, okay, to form the brick red precipitate of copper 1 oxide. But the first two are the best, all right, P1 and P2. Please tick this up, write this as your main points. 3 marks, okay, so any 3 points, they will go for 1, the prediction, and the explanation 1 and 2, all right. Point 3 is just an extra point. Now, E, classify the 6 substances below into 2 categories based on the size of their molecules. Classify them according to their size, because the smaller size one will be able to diffuse across the Visking tubing, and the bigger ones will be unable to diffuse across the Visking tubing. So you have to consider the size of the molecules, okay? Right, so what is your answer? Let's have a look here. Able to diffuse through Visking tubing will be glucose molecule, iodine molecule, fructose molecule, and amino acid molecules. These are all small in size. And unable to diffuse through the Visking tubing will be the sucrose molecule. Take note now, sucrose is a disaccharide and it cannot diffuse through the Visking tubing at all. Okay, starch molecule and protein molecules. These are even more complex. All right, starch and protein molecules. Now another example of classification. Classify the following membranes into 2 categories: selectively permeable membrane and non-permeable membrane. All right. So out of these 6, which one are selectively permeable? Now, egg membrane, that means the white membrane beneath the shell, that is selectively permeable. Plasma membrane of the cell is definitely selectively permeable. Okay. And nuclear membrane is also selectively permeable. That means they allow certain substances of smaller size, which contain molecules of smaller size, to diffuse through them. And those substances that contain molecules of bigger size cannot diffuse through the membrane. Okay, so Visking tubing is of course the one that we study here. Okay, but these are all the other examples of selectively permeable membrane. And for the non-permeable membrane, it's the eggshell itself, the rubber sheet, and boiled potato plasma membrane. Because boiled potato, if you boil the potato, the plasma membrane structure will be destroyed. Then it's not selectively permeable anymore. Okay. So that's all for this lesson or discussion. So all the best to you in your exams. Stay calm and study, and thanks for viewing. Please do share, like, and comment, and goodbye for now.